Okay, guys, welcome to Chapter 23. We're going to talk about blood vessels and circulation. Remember to be sure and watch the video podcast for Chapter 22 dealing with the heart because these chapters sort of move in synchrony. We're going to move from the heart to the blood vessels and to the blood. Okay, so basically, the circulatory system is a method for the body to deliver nutrients and oxygen to the cells that need it in order to generate ATP, which is the life energy for the body. This is how we're able to run the entire human system um, without having to have the cells immediately next to a source of oxygen or food. Uh, the circulatory system also manages to remove waste. So these are both vital processes in order to keep tissues and cells alive. We have to give them a constant source of oxygen and nutrients so they can have energy to perform life processes. And we also have to constantly remove waste products so that they don't build up and produce a toxic situation. The blood is the medium through which that happens, and the blood vessels are essentially the highways that carry the blood to and from their targets. So, if we're looking at blood vessels, they come in different flavors. Arteries are the vessels that always carry blood away from the heart. They tend to be rather elastic and rather large in diameter, and they're muscular, and this helps them distribute blood to different organs using a very important feature that we'll talk about in blood vessels, which is vasodilation and vasoconstriction. This is essentially a way for the blood vessel to change its diameter so that it can reroute blood to and from different organs. Arterioles provide distribution to capillary beds. Now, capillaries are the smallest of the blood vessels, but they're probably, in a way, some of the most important because it's in the capillaries where gas exchange actually takes place. This is where oxygen leaves the blood and enters the metabolically active tissue, and where carbon dioxide is picked up from metabolically active tissue. This is also where the majority of metabolic waste is removed from active cells and tissues to be brought eventually to the kidneys for detoxification and to the liver for detoxification. And then eventually that blood is going to make it back to the heart, specifically to the right atrium, where it will move through the pulmonary circuit, going from the right atrium to the right ventricle to the lungs, and then back to the left atrium, then to the left ventricle, and then to the entire body. The smooth muscle that's present in the arterioles also helps to regulate blood pressure. And how does it do that? It does that by either receiving increased or decreased what we call sympathetic tone. Basically, the sympathetic division of the autonomic nervous system sends messages to all of these vessels, the arteries, the arterioles, and even the veins to a lesser degree, to either contract or relax depending on the need for certain tissues in the body to receive a certain amount of blood in a certain period of time. Essentially, what happens when we have vasoconstriction is that blood pressure will increase because now you're making the container in which the blood sits smaller. And what happens when the vessels vasodilate is that the blood pressure will reduce. Okay, so we can control blood pressure doing this, and controlling blood pressure is one of the ways that we control blood flow. Essentially, the greater the amount of vasoconstriction, the less blood flow we're going to have to an area. The greater the amount of vasodilation, the more blood flow we're going to have to a particular area. So you could think of these as almost gateways to different regions in the body. The capillaries are thin-walled. They're even tinier than the arterioles, and a lot of diffusion takes place here. This is where a lot of nutrients leave the blood and enter the metabolically active tissue, and also where a lot of waste products are picked up. Okay, so it's these small vessels where we do a lot of exchange. The capillaries are well designed for this because they are so thin walled. You don't want to have a lot of thickness, a significant barrier between the blood and the tissue that it serves. Otherwise, diffusion isn't going to take place very efficiently. Okay, so the capillaries are designed to really ramp up the amount of exchange that takes place between the blood and the tissue fluid. Veins carry blood back to the heart. The venules basically are formed by a coalescence of the capillaries. The veins then run from the tissues back to the vena cava and then ultimately to the heart. Now, all blood vessels have three layers with the exception of the capillaries, and these three layers are the tunica externa, the tunica media, and the tunica interna. The arteries have a thicker tunica media. It contains a lot of elastic tissue and smooth muscle, while the arterioles um, are a little bit smaller than the arteries. They are where the control over blood pressure is exerted due to two factors. One, their small size initially, but also due to the fact that they have this smooth muscle in their middle layer that can engage in vasoconstriction or vasodilation. And again, remember, as these vessels vasoconstrict, the blood pressure will increase. As they vasodilate, the blood pressure will drop. Veins have a larger lumen and thinner walls, and they have valves to prevent backflow. Essentially, the blood in the veins is under such low pressure that the only way that we can get it back to the heart is through the assistance of the skeletal muscle that surrounds it. These are the muscles that you're familiar with, things like the muscles of the abdominal pelvic region, the pecs, the muscles in the lower legs, the hamstrings, the quads, and so forth. And in addition to that, the action of the diaphragm, which helps us breathe, also produces an oscillation in pressure that helps to move blood through the veins. But without these valves, we wouldn't be able to get the blood back to the heart, especially when we're referring to regions below the heart where we have to work against gravity. Venules are very thin and they have no valves present in them. And this is just a breakdown of a comparison between an artery and a vein. The artery is shown here on the left, the vein on the right. And the major feature that you're going to notice, the physiological feature, is the presence of these valves and also the difference in the relative thickness of these muscle layers. Notice again that in the arteries, what we have is a significant smooth muscle layer, a tunica media. We have a lot of elastic lamina between each of these layers, and this is so that the vessels have the ability to stretch as they experience pressure differentials as the blood is pumping through them. Essentially, the blood that is in these vessels is under relatively high pressure compared to the blood that's in the veins, and so a couple of things are important in order to accommodate that situation. You've got to have a vessel that is a little bit more structurally sound so that it can withstand the increase in pressure. In addition to that, this smooth muscle layer, again, is important in evening out the changes in pressure as the heart beats. Essentially, it allows the vessel to withstand the increases and decreases of pressure that result from the ventricles contracting because it has a little bit of contractile property of its own, and so it smooths out these big pressure differentials without causing now this vessel to rupture. The outer layer is basically epithelial tissue, and it is covered with a serosal layer again for lubrication. And then if we look over here at the veins, you can see the larger lumen, the tunica media, which is a lot less extensive, and again the outer layer, which is a basic epithelial layer. Okay, so you've got essentially, if we were to look at these tissue layers, let's look over here at the arteries. We've got epithelia, we've got connective tissue, okay, this elastic lamina. We've got smooth muscle tissue. We've got another layer of connective tissue. And then we've got another layer of epithelial tissue. Okay, so you've got different tissue types here, all acting in accordance with their designed function, right? Okay, let's talk a little bit about what some of these functions are. The arteries and the arterioles regulate blood flow, and this is primarily through sympathetic activity to the smooth muscle that's in the tunica media. Essentially, what we have here is something called sympathetic tone, and it's one of the things that helps maintain our blood pressure. If we didn't have this sort of background level of vasoconstriction at all times, what would happen is that our blood pressure would fall so low that we'd pass out and perhaps die. Okay, so this is one of the interesting background functions of the sympathetic division of the autonomic nervous system that keeps a sufficient amount of blood flow and pressure to the active tissues, again, to keep the entire body operating, right? This allows us to get enough oxygen and nutrients to the tissue so that we don't end up starving the tissue for energy, but also we don't neglect again to remove the waste products. Decreased sympathetic activity or nitric oxide causes relaxation, also known as vasodilation. So basically the difference in the diameter of these arteries is determined by the amount of sympathetic innervation that occurs. There is no parasympathetic role in this aspect of circulation. The arterioles adjust the flow into the capillaries, and they do that using special smooth muscle structures called capillary sphincters, which essentially can either shut off or open up an arteriole to allow a capillary bed to perfuse. The capillaries are the sites of gas exchange. This is where oxygen is delivered to active tissue and carbon dioxide is removed, in addition to delivering nutrients and picking up waste products. The systemic venules in the veins serve as blood reservoirs, and they hold about 64% of the total blood volume. So what you're looking at here is just the structure of a capillary, and what I want you to notice is that there's a fundamental difference here in this vessel versus the other two that we've looked at, and that's the fact that you've got virtually no tunica media. You've just got an endothelial layer, and then you've got some basement membrane, some connective tissue. And again, that is to affect gas exchange and diffusion. We don't want to have a significant barrier between the blood that's in these vessels and the tissue that surrounds them. Capillaries are composed of endothelia. They have very thin cells, and this allows for easy diffusion. The cell nuclei protrude into the lumen, and they're connected from arterioles to venules in networks. They sometimes have a direct route from an arteriole to a venule. Capillary filling is controlled by small arterioles and precapillary sphincters, and this is under the control again of smooth muscle. This autoregulation is basically the ability of a tissue to adjust blood flow into the area according to demands. And what would these demands be? Well, a lack of oxygen would be an example. A lack of nutrients would be another example. Okay, so these are all possible homeostatic changes that could result in a need to either increase or decrease the amount of oxygen and nutrients going to a particular area. And the body will adjust according to that. 50, pick your favorite 30. Okay? Okay, and what you're looking at here is just a diagram showing you a perfused capillary bed. You can see here the blood flowing into the arterioles, and in this case the capillary sphincters are wide open, and you can see that the blood is flowing through all these different capillaries, and as a result now we're having a significant amount of oxygen dropped off and carbon dioxide picked up, in addition to the diffusion of nutrients and waste products that we discussed. Again, all these capillaries eventually coalesce back into a venule, which will eventually enter a vein, which will eventually feed into the vena cava, which will uh, eventually get back to the heart. This is what it would look like in the event that we had now the capillary sphincters closed and the demand for oxygen and nutrients was reduced. Okay, so you can see here that we can dramatically affect now not only the rate at which we deliver oxygen and nutrients to tissue, but also um, the blood pressure. Basically, when the capillary beds are wide open, that's going to result in a drop in local blood pressure, whereas if the capillary beds are shut off because these sphincters are closed, you're going to have an increase in the blood pressure in that area. Okay? Essentially, the larger the container becomes for the fluid that's in it, the lower the pressure is going to be. Okay, capillary exchange is essentially the swapping out of oxygen for carbon dioxide and of waste products for nutrients that occur in these very thin-walled vessels. The slow rate of flow in the capillaries allows this to happen. In addition to that, remember that the capillary walls are very thin, and as a result, diffusion happens a lot more readily in this sort of setup. This allows time for exchange through the walls of these vessels. Blood pressure pushes components out of the capillary. It permits filtration of fluid out of the capillary, and this happens mostly in the first half of the capillary network. Now, something you have to keep in mind here, it's very important. This is an aspect of this chapter we could call hemodynamics, and that's just a fancy way of saying what are the physical processes that govern which way components are going to move when they're in these vessels that are serving now all this metabolically active tissue. And it comes down to either osmotic pressure and or hydrostatic pressure. Now, what is osmotic pressure? That's just the flow of water down its concentration gradient from where it's most concentrated to where it's least concentrated. Diffusion is another aspect of this. Basically, the way that molecules will move in the absence of active transport is away from the area where they're most concentrated in an attempt now to equalize concentration on either side of a tissue layer or of a membrane. Okay, so the two go hand in hand. Essentially, where the solutes go, where the dissolved molecules go, the water will follow. Okay, and the water is the major component that's going to determine the blood pressure. So you have to consider that when you're talking about the blood and the tissue fluid. Basically, in the tissue fluid and in the blood, you have dissolved components. You've got ions, you've got proteins, you've got waste products, you've got gases. All of these are suspended in essentially a liquid matrix. And where the water goes is going to be determined in part by the relative concentrations of those solutes in the water. Basically, if you've got a solution that has a lot of stuff dissolved in it and it's separated by a semi-permeable layer from another tissue compartment that has fewer solutes in it, what will happen is that the water will move from the area where there's fewer solutes to the area where there's more solutes in an attempt to dilute what's on the other side of that barrier. Okay, and that's what we call osmotic pressure. A nice way to remember this is that water chases solutes. So where electrolytes go, where protein goes, where solutes go, the water will follow. So remember that in the blood plasma, you have electrolytes, you have proteins, you have dissolved gases, and all of these are going to exert an osmotic pressure. And the same is true in the tissue fluid. In the tissue fluid, you also have proteins, electrolytes, and dissolved gases. It turns out that in the blood, you have a little bit more of that osmotic pressure than you have in the tissue fluid. So that's going to tend to draw water into the circulatory system. Now, the other thing that's at work is the hydrostatic pressure. Now, what is the hydrostatic pressure? That's just the physical force of the fluid on the container that it's in. And when we're talking about the blood, that physical force is the result of two factors: the pumping action of the ventricles, and also the degree of vasoconstriction or vasodilation in the vessel that contains it. Okay, so that's something that we can modulate. And what we're interested in when we think about where fluids and solutes go is we're interested in the net pressure. What is the combined effect of these two forces? Okay, and in the first half of the capillary bed, the combined effect, when you take into account hydrostatic pressure and osmotic pressure, both of which work in both directions, the net effect is for filtration to occur out of the capillary bed in the first half of the capillary network. Colloid osmotic pressure is that osmotic pressure that pulls fluid into the capillary. This is produced primarily in blood plasma by the large proteins that are in there, the major one being albumin. Serum albumin is a large molecule, and it doesn't diffuse easily across cell membranes or across endothelial tissue, so it tends to sit there in the blood plasma and it tends to pull water towards it. So this causes reabsorption of fluid from outside the circulatory system to inside the circulatory system. The balance of blood pressure and osmosis determines the amount of fluid that's in circulation. Excess tissue fluid is eventually returned to the circulatory system through the lymphatic system, and local signals can adjust capillary flow in addition to these other factors that we've discussed. So essentially, when we're looking here at capillary exchange, this is sort of a nice diagram. What you're seeing here, this is the blood coming to the tissue from the heart, right? And so what happens here is blood flow from the blood arterial into the capillary, and the net osmotic pressure, both in and out of the vessel, tends to force filtrates and components out through the endothelial layer and into the surrounding tissue. So what does this mean? This means that we drop off nutrients, we drop off oxygen to tissues that need it. Then as we move through the capillary bed, something interesting happens. And what happens is that the blood pressure drops. And the reason that it drops is because the blood basically is perfusing a very large area at this point, and it's meeting a lot of resistance. And so what happens here is that blood pressure and blood flow tend to decline, and that little trick, okay, now tips the scales at the venous end of the capillary bed towards reabsorption. So what we tend to do here is pick up waste products, pick up carbon dioxide, load that into the blood. And then what happens here is that blood flow here from the capillary bed enters venules, which eventually enter veins, which eventually feed back into the vena cava, which eventually take it back to the heart. Now, what happens to a lot of this filtrate? Well, some of it contains components that the tissue cells use, and the extra tissue fluid enters the lymphatic system. Now, we're going to talk a little bit more about the lymphatic system a bit later on, but essentially lymphatic capillaries are similar in many respects to veins, in that they have relatively thin walls, they contain valves, and they don't really have much of a driving force other than skeletal muscle to push the fluid in them through the lymphatic system. The purpose of getting the fluid into the lymphatic system before we return it to the circulation, as shown here, is to clean it of pathogens. Basically, even though you've got all these wonderful defense systems in place, you've got your nonspecific barriers, you've got the immune system, you've got the inflammatory response, all those things that try to keep us safe from disease. Occasionally what will happen is that pathogens will make it into the tissue fluid, into the blood. And what we want to do is check for their presence by sending this fluid through special lymphoid organs such as the spleen, the tonsils, the Peyer's patches, and in the case of the lymphatic system proper, the lymph nodes. And long story short, what happens in these organs is that the flow of tissue fluid drops to the point where in these organs, the lymphocytes, the macrophages, and um, the reticulocytes get a chance to really examine the tissue very thoroughly for the presence of pathogens. And if they're detected, they'll be engulfed, and that bubble that contains the disease-causing entity will then be fused with a lysosome, which contains these powerful digestive enzymes, which will then chop the pathogen up into little pieces. And then those little pieces will be presented on the surface of these cells as a warning signal to the immune system that we have trouble in town. Essentially, a disease-causing entity has invaded our space. And what this does is it serves as a way to recruit T and B lymphocytes that are going to specifically battle that particular pathogen and take him out using a combination of chemical attacks that are designed to destroy him. Now, once that fluid has been cleaned, it's returned back to the venous circulation at the right and left subclavian veins up here under your clavicle. And at that point now, we've protected ourselves in yet another way from the presence of disease. So this is an important role for the lymphatic system. Now, if we impede the flow into the lymphatic system, and there are diseases in which this happens, one of them is a condition known as elephantiasis, where parasitic worms can actually get into the deep tissues of the body and lodge themselves inside these lymphatic capillaries and impede the flow of fluid from around the tissues into the lymphatic system. What this results in is massive tissue edema. So the tissues will distend, they'll fill up with fluid, with water, and they'll get very large and stretched out. And eventually what will happen is that you'll end up here with extremely distorted tissue compartments. Okay, for instance, if you have these parasites in the inguinal region, in men this results in extreme swelling of the scrotum. If you have it in the popliteal region, the lower leg may swell up. Just to give you a few examples. Okay, so this is what we would call peripheral edema. Okay, venous return. Now, how do we get the blood back to the heart? Blood enters the veins at very low pressure, which is inadequate to overcome the gravity, especially below the heart, and return the blood to it. So what happens is, as a result of skeletal muscle contraction, we squeeze the blood in the venous system through these one-way valves, and eventually it climbs all the way back to the heart. Because of the venous valves, the flow is always in one direction, and that's towards the heart. Now, occasionally what can happen is that these valves can weaken because of the increased pressure, and they can buckle. And if they do that, what will happen is that you'll generate something called a varicose vein. Now, a varicose vein isn't just unattractive and uncomfortable, it's also dangerous because it represents a site where blood flow is decreased. And remember that any time blood flow slows down, blood can spontaneously clot. And if that happens now, what we can generate are traveling masses called emboli, which can eventually lodge in narrow vessels such as capillary beds and keep blood from flowing to anything downstream of that. And that's bad because that can result now in tissue death, necrosis. If this happens in the heart, it can cause a heart attack. If it happens in the brain, it can cause a stroke. And so one of the things they do for people with varicose veins is they recommend, for instance, if you work at a job where you have to stand for long periods of time, that you try and move the lower limbs, okay, to work the skeletal muscle pump to keep blood flowing back to the heart. They also recommend pressure hose as a way to, overcome the pull of gravity away from the heart in this case, so that we can get adequate venous return. And in the event that you have a thrombosis, basically a block in a varicose vein, they can remove the vein using surgical procedures such as lasers, which can actually vaporize them. And the body has an amazing ability to rebuild blood vessels after they've been damaged in a process known as neovascularization. And basically what it'll do is generate now a bypass to go around the surgical intervention. Okay, so even though you might lose a section of a vein, your body can grow it back after a time. Okay, what else moves blood back towards the heart? The respiratory pump. Basically, when you breathe, the action of your diaphragm decreases and increases thoracic pressure, and what this does is produces rhythmic contractions that press on the vena cava and help to draw blood back to the heart. Exhalation allows the refilling of the abdominal veins. Here you can see the skeletal muscle pump in action, right? Here we are, way down in the lower extremities of the body. We're looking here at a lateral view of the calf, and what you can see here is that you've got these valves that are far removed from the heart, and then as we squeeze, as the skeletal muscle pump squeezes, we force blood through these valves, and it begins to climb one valve at a time, eventually making it all the way back to the iliac veins, and then eventually to the inferior vena cava, and then eventually all the way back to the right atrium. And it's this squeezing action and the valves that work together to allow this to occur. Okay, what are some of the factors that govern the direction of blood flow? And it's basically the principle that nature abhors a vacuum. Essentially, things always move from an area of high pressure to an area of low pressure. They can't help it. So they move down their pressure gradient. The greater the gradient, the greater the difference in the pressure between two compartments, the more rapid the flow is going to be. Blood pressure is highest at the aorta at around 110/70 millimeters of mercury. Remember that top number is the systolic and that bottom number is the diastolic pressure. The reason that we can take a pulse is because of these big pressure differentials that we see in arteries. Arteries in general are closer to the heart, and as a result, in areas of the body where the arteries happen to run close to the surface and there's not a lot of overlying fat, it's easy for us to take a pulse rate and to get some idea of how rapidly your heart's beating. Okay, so you can't take a pulse in a vein because of the pressure drop that happens after we move through those capillary beds. Blood pressure declines as it flows through more vessels, and the reason for that is that it encounters resistance, generally from rubbing against the sides of the vessels. That's like backflow. Okay. In the arterioles, you see a major drop in blood pressure due to smooth muscle contraction, which results in vasoconstriction. In the capillary beds, we only have a pressure of between 35 and 16 millimeters of mercury, and at the venules, it's about 16 millimeters of mercury and 0 when we get back to the right atrium. So if you look at this chart, you can see how blood pressure changes as we walk away from the heart. So imagine in this graph that we're proximal to the heart. Okay, we're very near the heart here. Notice the pressure changes in the aorta. Notice how extreme they are. We're basically rocking up and down from between about 110 to as low as around 70. Okay, and the reason for this is we're very close to the pump at this point. We're very close to the ventricles. As we move away from the heart, what happens is that we experience resistance because of the drag against the vessels. The other thing that begins to happen is that the vessels start to get smaller. And the smaller they get, the greater the drag. And the reason for that is something called laminar flow. And basically, the best way to describe laminar flow is that if you've got a liquid or a gas, the way that it moves is in layers, okay? So you can imagine a volume of air or a volume of liquid, and essentially what you'll see in terms of molecular motion is that these things will move in uniform layers. Now, what happens in laminar flow is that if we impede the movement of one of these layers, we slow it down, then we create a situation that results in drag. So if we imagine here that this is the wall of a vessel. Okay, I'll just remember, this is my very bad drawing here of a vessel. Okay, so this is a vessel. This is the blood flowing through the vessel. All right, and you can imagine here that the fluid that is near the periphery is gonna slow down because it's going to drag against the walls. And what happens here is that the fluid in the middle will experience less drag. Okay, so what does this tell you? Okay, this tells you that the net speed of a fluid as it flows through a large vessel is going to be greater than the net speed as it flows through a tiny vessel, because in a tiny vessel much more of the liquid is going to be making contact with the walls than it is in a big old pipe. Okay, so you can imagine, just think in terms of like a hose. Okay, it's easier for water to flow through a hose than it is for it to flow through a little tiny piece of tubing, because it's got to overcome all that drag. Now notice what happens as we move away from the heart. We head away from the heart and we experience now a decrease in overall pressure and also a decrease in the fluctuations of pressure. Okay, so down, down, down we go from the arteries to the arterioles, and then when we hit the capillaries, look at this big drop off here. Okay, and the reason is because now what's happening is that we're entering a huge number of vessels with a big cross-sectional area, and also a major amount of drag because the vessels are also small. And what happens as a result is the backflow goes way up, and the overall pressure goes way, way down. Okay, um, and now what happens by the time we enter the venules is that we have virtually no pressure at all. And so the only way that we can get blood back to the heart is through that skeletal muscle pump and through the action of breathing. Now look what happens as we move from the venules to the veins, and we get all the way back here to the vena cava. See that little spike right there? That little blip? That is the draw that's coming from the beating action of the heart. Once we get back close to the heart again, because you have to remember that this whole chart is basically running in a circle here. We're leaving the heart, we're heading out to the body, and then we're heading back to the heart. Okay, and by the time we get to the vena cava, what's happening now is that as the ventricles are dilating and contracting, they're giving us a little bit of draw. Okay, so we have a little bit of that effect here at the very tail end of the venous system. All right, so hopefully that makes a little sense to you. Factors that regulate blood flow and blood pressure, basically blood volume and ventricular contraction are responsible for cardiac output. This is under the control of the cardiovascular center of the hypothalamus. Okay, there's also vasculature resistance, which is the opposition to flow, and this is largely dependent on lumen diameter and vessel length, as well as the viscosity of the blood. Now, some of these you've probably heard before, some of these probably are new to you. The lumen diameter, what's that? Well, that's how big the hole is in the pipe. Okay, basically the greater the diameter, the less resistance. The smaller the diameter, the more resistance. The vessel length, the longer the vessel is, the greater the resistance to flow, because the greater the amount of drag going through that pipe. And then finally, blood viscosity. Now you might think, viscosity, what is that? I've never heard that term before. Viscosity is a term that means resistance to flow. Basically, an extremely viscous solution would be something like maple syrup, and a solution that's not viscous would be something like pure water or even alcohol. Okay, so as blood viscosity changes, as the amount of formed elements and the amount of solutes in the blood plasma goes up, the viscosity goes up, the resistance to flow goes up, and the result is that the rate of flow goes down. So smaller lumen occurs with vasoconstriction. This leads to greater resistance. The greater the vessel length, the greater the resistance, and the higher the viscosity, the greater the resistance. Okay, the cardiovascular center is located in the medulla oblongata. It's part of the brainstem. And what it does is it regulates our heart rate and our stroke volume, as well as our blood pressure and the amount of blood that flows to specific tissues. The mechanisms are both neural and hormonal. Okay, in order to inform the brain that the blood pressure should be changed, we have input that comes from different parts of the brain. We've got the cerebral cortex, which provides now a conscious input to the effect of blood pressure. The limbic system, which is the emotional input to blood pressure. The hypothalamus, which detects changes in temperature or blood volume and adjusts blood flow accordingly. Okay, so this is below our conscious awareness. The limbic system and the cortex are within our conscious awareness. We also have input from sensory receptors and nerves, such as the proprioceptors, the baroreceptors, and the chemoreceptors. Now, what are each of those? Well, your proprioceptors are those pieces of nervous tissue that inform you of where your body is in three-dimensional space. Okay, so as you move, as you sit, as you stand, your proprioceptors tell you essentially what's going on, where you are. Baroreceptors detect pressure changes, and chemoreceptors detect changes in blood chemistry. And the two most important components that affect chemoreceptors are carbon dioxide and oxygen concentration, as well as pH. Okay. Proprioceptors monitor the movements of joints and muscles. This causes an increase in heart rate as exercise begins, and as a result increases cardiac output, which increases blood pressure. Now, why does all that make sense? Okay, well, think about it. Let's say that you're going to do 50 push-ups. Okay, um, you start out not doing anything, then you drop on the floor and you start to pump yourself up and down, up and down, up and down. Well, what's happening? Muscles are contracting. As muscles are contracting, what are they doing? Well, they're using energy. Okay, they're using glucose, they're using oxygen. Okay, and in order to pay that debt, what we have to do now is increase our cardiac output so that we increase the rate at which we drop off oxygen and nutrients to the muscle, so that the muscle doesn't just quit on us. Okay, and this net result is to increase blood pressure. Okay, so we can increase cardiac output by increasing stroke volume and increasing heart rate. Okay, and this can go to increase blood pressure. Baroreceptors in the aorta and the carotid bodies jump into action if your blood pressure drops. The aorta, remember, is that large vessel that comes straight out of the left ventricle and delivers blood to the rest of the body. And the carotid arteries are found in the neck, and they supply oxygenated blood to the brain. If your blood pressure drops, we see an increase in sympathetic stimulation, which increases the cardiac output and increases the blood pressure. Now, think back to when we talked about the endocrine system, and remember that the sympathetic division of the autonomic nervous system has a nickname, and that's the fight or flight division. Okay? So think about what would be important for you to get out of an emergency situation. And what would be important would be for you to increase the amount of oxygen and fuel delivered to your muscles, increase your peripheral blood pressure, increase the amount of fuel that's in the blood by increasing the blood glucose concentration, and increasing your respiratory rate so that you can load more oxygen into the blood more quickly. Okay? So all this happens under sympathetic stimulation. We increase the cardiac output, we increase the blood pressure. The other thing that we increase is the respiratory rate. Two other things that jump up are the rate of gluconeogenesis and glycogenolysis. Remember that gluconeogenesis literally means making new sugar, and so the reason we do that when we're under sympathetic stimulation is because we want to jack up blood glucose so we can deliver more fuel to the muscle. Okay, and glycogenolysis is just the cleaving of those long glucose polymers, glycogen, into glucose molecules so that we can get that into the blood and deliver it to the muscle as well. A decrease in parasympathetic stimulation also results in an increase in cardiac output and an increase in blood pressure. Okay, so these two guys work together. There's also chemoreceptors that are present in the aorta and in the carotid body. Basically, if you experience low oxygen concentration or high carbon dioxide concentration in the blood plasma, or high acidity, we will increase resistance by increasing vasoconstriction and increasing blood pressure. And another thing that happens again is we increase the respiratory rate. Now, it probably makes total sense to you why low oxygen would trigger this, right? If there's low oxygen to a tissue, what do we need to do? Get more blood to it, and the blood that we're getting to it needs to have a higher oxygen content. High carbon dioxide, what does that mean? Well, that means that the blood isn't removing the waste gas fast enough in order to keep the tissue healthy. Okay, so what are we going to do? We're going to increase cardiac output, and we're going to increase heart rate and also respiratory rate so that we blow off the waste gas. But the high acidity might confuse you a little bit. Why would high acidity result in an increase in blood pressure, increase in cardiac output, and increase in respiratory rate? And the reason for that is a little bit not so obvious. What you have to remember here is that when carbon dioxide is produced by cells that are undergoing respiration, remember that glucose is broken down into carbon dioxide and water, okay? And the other products are massive amounts of ATP and heat. But as this carbon dioxide is built up in active tissue, what happens is it combines with water and it forms a molecule called carbonic acid. Okay, so I'll draw this chemical equation down here for you. Okay. Now, carbonic acid, H2CO3, can dissociate in water, forming now free protons, CO3 there. And when you've got free protons in solution, what happens to the pH is that it drops, so the solution becomes more acidic. So when we say we have an increase in acidity, we're sort of in a backhanded way saying we're increasing the amount of carbon dioxide in the tissue, because this equation is basically an output. This is the carbonic acid. These are the protons. This is what lowers the pH. We're basically saying that we're forming more carbonic acid, which is now dissociating here into H plus and bicarbonate, HCO3 minus, and it's the H plus that lowers the pH. Finish out the equation here. Okay, so this is an equilibrium. Okay, these reactions will go in either direction, and the direction that they go in is going to be governed by the concentrations of what's on either side of these arrows. Okay, but essentially in metabolically active tissue, what's happening is that we're loading up on this, and as a result, we're producing lots of that. Okay, so what do we have to do in order to relieve this stress? Is we have to get rid of the carbon dioxide. Okay, so what happens is we increase the rate of blood flow, we sweep away the CO2. As a result, now the equilibrium shifts back in the other direction, goes to the left. The protons are consumed, and the pH begins to rise again, go back towards 7. And this is just a graph showing you all these various inputs. Okay, higher brain centers, and then the proprio-baro and chemoreceptors, how they all feed in here to the cardiovascular center in the medulla. And then the outputs are via the vagus nerve. Remember, that's the 10th cranial nerve, which feeds to the heart. The cardiac acceleratory nerves are the sympathetic leads to the heart. And the vasomotor nerves are the vasoconstrictors. Okay, so these are the sympathetic outputs, the sympathetic leads, and these are the parasympathetic leads. Okay, so we have a way to either increase or decrease the heart rate depending now on the body's needs at the moment. Okay, okay, let's talk about these nerves to the heart. The autonomic nerve leads to the heart. The sympathetic leads are basically there to increase the heart rate and the force of contraction, and to increase the cardiac output, which leads to an increase in the blood pressure. The parasympathetic leads basically do the opposite. Now, the parasympathetic leads, that's primarily the vagus nerve, okay? And the sympathetic leads come from a few different places, okay? There are some paravertebral ganglia that have direct leads to the heart. But remember from the chapter on the heart that the heart can basically beat on its own. It doesn't need any nervous system input in order to pump. But it does need nervous input in order to know how quick to pump. So as we increase or decrease our activity levels, it's these leads, these sympathetic and parasympathetic leads, that talk to the SA and the AV node and either speed up or slow down their intrinsic rhythm. Okay, so these are basically suggestions to the electrical tissue that's already embedded in the heart wall. Okay, vasomotor nerves lead to arterioles, which cause smooth muscle to contract. This increases vasomotor tone, which increases vascular resistance, and the result is to increase blood pressure. The leads to the veins cause them to contract their smooth muscle and to move more blood up towards the heart, and this also helps to increase blood pressure, because if you increase the amount of blood delivered to the heart, what you're doing is you're increasing what we call the preload. Okay? The preload is basically the amount of blood flowing into the first of the four chambers. The greater the amount of blood you throw into the heart, the more blood you're going to pump out of the heart, because the amount of blood that enters is always going to be equal to the amount of blood that leaves. So the greater the preload, the greater the cardiac output. That's just an intrinsic property of the heart. And so this is one of the reasons why when you exercise, your cardiac output goes up, okay? Because you're delivering more blood to the heart, and as a result, with each beat, you're sending more blood to the rest of the body. Okay, how do hormones act? Well, the renin-angiotensin-aldosterone system is probably the most important hormone system that affects blood pressure and blood flow. Basically, angiotensin 2 is a potent vasoconstrictor. So what it does is it raises blood pressure. Aldosterone, which is generated by the adrenal cortex, causes the kidneys to retain sodium, and as a result now the water will chase the sodium back into the body, and this will result in an increase in blood pressure as well. Epinephrine and norepinephrine increase cardiac output and blood pressure. ADH, also known as vasopressin, is our water retaining hormone. Remember that it acts on the collecting ducts in the kidneys to change their permeability to water. Essentially, when you've got a lot of ADH around, the collecting ducts are very permeable to water, so it's very easy for us to retain a lot of what normally would be lost in the urine. This, as a result, eventually increases blood volume and increases blood pressure. This also triggers thirst, so we seek out water. This, along with water retention in the kidneys, serves to increase blood pressure. Atrial natriuretic peptide has the opposite effect. Basically, it triggers vasodilation and loss of sodium and consequently water in the urine, and as a result, blood pressure tends to drop. Okay, so if we look here at the overall diagram, you can see what the initial triggers are and how the feedback mechanism works. Best advice I have for you is check with your book here, because to be honest with you, this is a little bit too small for me to even pick out what's on here. But essentially what happens here is we have a change in blood pressure. Let's say we have an increase or a decrease in blood pressure. This can lead to now an increase in sympathetic stimulation to the heart. The heart rate will go up. This will increase the cardiac output. At the same time, what can happen is that we can produce angiotensin 2, which is a potent vasoconstrictor. This also helps trigger the release of aldosterone, which increases blood volume. Once blood pressure and blood volume go back up, what happens now is we have a negative feedback loop that cuts this entire initiating signal out, okay, and now we quit the secretion of the renin and angiotensin and aldosterone. Okay, one of the things that you do as a health professional is that you check somebody's pulse rate in order to get some idea of what their heart rate is under various levels of exertion. Your pulse in your arteries is essentially equal to your heart rate. What you want to do in order to take a pulse is to press an artery against a bone or muscle. Sites that are used include the radial artery, which is on the thumb side of the wrist, the carotid artery in the neck, the brachial artery in the arm. And what do all these sites have in common? They all have arteries that run very close to the surface of the body, so you can feel those changes in pressure much more effectively. Remember, you cannot take a pulse in a vein. A couple of conditions that you may run into when you do this are tachycardia, which is a rapid resting heart rate of greater than 100 beats per minute, or bradycardia, which is a slow resting heart rate of less than 50 beats per minute. We can also measure the blood pressure, and the reason that it's important to measure your blood pressure is to detect whether or not your blood pressure is on a consistent basis either too high or too low. The device that we use here is called a sphygmomanometer. And we generally use it on the brachial artery. That's the artery in your arm, your upper arm. We inflate the cuff to raise the blood pressure above systolic pressure. So essentially we cut the blood flow off temporarily. And then we let the pressure out very slowly until, using either a visual cue as a result of the dial that's affixed to the blood pressure cuff, you start to see a pulse or jump. Okay, the first place where you start to see it kick, that would be the systolic pressure. Or if you listen through a stethoscope, if you have good ears, you can hear the periodic whooshing sound coming through the vessel. That will be your systolic pressure. That's your high pressure. That's the pressure that's generated when the ventricles are squeezing. Okay, so this is your systolic pressure, and then you continue to release the pressure on the cuff very slowly until those pulsations go away, either visually or through the scope, and that's your diastolic pressure. Normal blood pressure values are 120 millimeter for systolic and 80 for diastolic. If you've got any graded. Yeah. And I'll have these up tonight. Okay? Thank you. Good luck. It was a pleasure having you in my class. Okay, there's two main circulatory routes in the body, the systemic and the pulmonary. The systemic is basically the blood going to everywhere but the lungs, and the pulmonary is the route to and from the heart to the lungs. Systemic circulation is where oxygenated blood travels from the heart throughout the body, losing oxygen as it goes, because what is it doing? It's delivering that life-giving gas to the peripheral tissues. All systemic arteries branch from the aorta. All systemic veins empty into the superior vena cava, inferior vena cava, or coronary sinus. Deoxygenated blood then returns to the heart. And this is just a diagram showing you the pulmonary circuit, which is here. Okay, notice here, right atrium, right ventricle, to the lungs, and then back to the left atrium. And then we enter the systemic circulation, the left ventricle to the aorta to the rest of the body. Okay, so when we're talking here about the blood flow to and from the heart from the lungs, we're talking again right ventricle to the lungs, back to the left atrium. Systemic circulation is left ventricle to the body to the right atrium. Okay. Now, something to notice about the circulatory routes. Okay. In the systemic circulation, remember that oxygenated blood is going to be found in the arteries, and deoxygenated blood is going to be found in the veins. Okay, that's the route to and from the body. In the pulmonary circuit, this is reversed. In the pulmonary circuit, deoxygenated blood is found in the arteries, and oxygenated blood is found in the veins. Now, you might say, well, why is that? Well, it's because of the direction of the blood flow. Remember that arteries always carry blood away from the heart. Okay, so the aorta is the major systemic artery leading right out of the left ventricle. The pulmonary trunk and the pulmonary arteries are the major arteries leading away from the heart out of the right ventricle. But notice that the blood in the pulmonary trunk and the pulmonary arteries is blue. Why is that? Well, because all the oxygen was lost as it was dropped off to the rest of the body's tissues. Now what happens in the lungs is that that blood will pick up oxygen from the air that you breathe. The color will change from blue to red, and then it will flow back here to the left atrium, and now it will enter the systemic circulation as it goes into the left ventricle and then heads out the aorta to the rest of the body. Okay? This is just showing you some of the common circulatory routes. Now, the major thing to remember here is just that if you remember your names for your body regions, then you can essentially name these arteries and veins. Okay, so I can give you a few easy landmarks here. Okay, let's go with the systemic circulation. This is the aorta. Okay, as the aorta heads up, it's called the ascending aorta. This is the aortic arch. This is the descending aorta. In the region of the chest, it's called the thoracic aorta, and as it passes through the diaphragm, it now becomes the abdominal aorta. Then what happens is that it splits, and it splits right before it leaves the abdominal pelvic cavity into the left and right common iliac arteries. Those left and right common iliac arteries then branch into the internal iliac arteries that project medially and posteriorly, and the external iliac artery that projects now more superficially. The external iliac artery then becomes the femoral artery, okay, up here near the femur, okay. So femoral artery runs near the femur. As it passes behind the knee, it becomes the popliteal artery. Remember that the back of the knee is called the popliteal region. And then, as we enter the lower leg, we have an anterior and posterior tibial artery. Well, guess what bone they run next to? The tibia, okay? And then you've got on your lateral strut the fibular artery, okay? And that runs next to the fibula. Okay, so again, you can use the bones and the regions of the body as major landmarks. Now, if we head up back here to the aorta, okay, as we branch off here, what you've got now is the brachiocephalic trunk on the right side, and you've got on the left side the right subclavian and the left carotid coming right off the aorta. So one of the things that you'll notice is that in the systemic circulation, not perfectly symmetrical. Okay, the right side and the left side are a little bit different. The brachiocephalic branches into the common carotid and the subclavian. Okay, the subclavian, as it passes under the arm, becomes the axillary artery, and then as it enters now the upper arm, okay, it's the brachial artery. Well, what is the name for the region of the upper arm? The brachial region. Okay. Then what happens is we pass the elbow, we become now the antecubital artery as we pass right in front of the elbow joint. This is a major site for drawing blood. And then as we pass down into the forearm, you see three arteries. You see a radial, a medial, and an ulnar artery. Okay, and then when we get to the hands, we have the superficial and deep palmar arteries, and then we have the digital arteries down here. Okay, so you can use a lot of these regions of the body as a way to figure out the names for each of these vessels. We can talk about some of the major vessels in the body. The feeds to the brain, the major ones are the common carotid, which branches into the internal and external carotid arteries, and this takes blood to the brain. Okay, this is just a zoom up here on the brachial region. Okay, so what are we looking at here? There's the aorta. Okay, this is now the subclavian. Okay, right up here over the top of the clavicle. Okay, and then it drops below it. So subclavian below the clavicle. Then it becomes the axillary artery as it dips down here right around the glenohumeral joint. Okay, and then we continue. We become now the brachial. The brachial then becomes eventually the right radial and ulnar, and then there's also a medial artery in there, and then the superficial and deep palmar arches. Up here, you're looking at some of the major feeds to the brain. Okay, again, here's the clavicle, major landmark. There's the brachiocephalic trunk. There is the common carotid, the right common carotid, and here is the right subclavian. So carotid to the brain, subclavian to the arm. As it passes under the clavicle, it becomes the axillary. Okay, and then if we follow now um, the carotid, okay, walk up to the carotid, it branches into the external and the internal carotid. The internal carotid breaks medially and posteriorly. The external carotid breaks laterally and more anteriorly. Okay, as we head into the brain, okay, other major branches include the cerebral, okay, the basilar, which basically runs almost really close to the brain stem. Okay, and then other branches that come off the external carotid include the middle cerebral. Okay, you've got facial arteries in here. You've got mandibular arteries. Another major branch that comes off the brachiocephalic trunk after we bust into the carotid. The next thing that comes off is the vertebral artery. Okay, and the vertebral artery passes through these transverse foramina. Okay, um, and this is one of the distinguishing features of the cervical vertebrae. Okay, is the fact that they contain these transverse foramina that are not oriented um, laterally, they're oriented horizontally, okay, and that's to accommodate the vessel that runs through them. No other vertebrae in the spinal in the vertebral column has that feature. Okay, so that's one of the distinguishing features. What you're looking at here is the underside of the brain. Okay, there's the optic chiasm. Those are the optic tracts. These are the optic nerves. They've been cut here so that you can see the olfactory tracts right here. Okay, these are the mammillary bodies. All right, and this would be essentially the pituitary gland. There's the pons. Okay, there's the basilar artery running right across the midline of the pons. This is the medulla oblongata. Okay, and this whole structure here is called the circle of Willis, the arterial circle. The circle of Willis has several components to it. It's got the anterior cerebral and anterior communicating arteries right there. There's the anterior communicating. You've got the internal carotids feeding into here. This is the posterior communicating. Okay. And then you've got the basilar artery feeding into here. Okay. Basilar artery runs up here and joins now the posterior cerebral. Okay. And this whole thing is it's like kind of like a roundabout. I don't know if you've ever seen a roundabout um, as a traffic pattern. Okay, basically it's a intersection where the road goes in a circle, and you've got exits that come off every part of the roundabout. Well, it's very similar in a sense to the purpose of this particular um, arterial structure, because what we're doing here as a result of having these anterior and posterior communicating arteries, there's a posterior, there's the anterior, okay, and then you've got here the internal carotids jacking in right here, the posterior cerebrals, okay, is that we're ensuring blood flow to the brain in the event of a blockage anywhere else along um, these other arteries. Basically, if we have a block, say, in the posterior or the anterior communicating, we can still get blood to the other parts of the brain by running through the rest of the circle through the bypass, okay, and then we can still get blood to the temporal lobe, to the frontal lobe, etc., to the brainstem, and so forth, because we've got this sort of bypass set up, okay. Okay, if we look here at the roots that run through the pelvis and through the lower limb, um, basically you've got here the abdominal aorta, there's the common iliac. The internal iliac, remember, is going to break medially and posteriorly. The external iliac is going to break laterally and more superficially. It then becomes the femoral. Then it descends behind the knee, becoming the popliteal. And then down here in the lower leg, it breaks into the anterior and posterior tibial, which went on either side of the tibia, and the fibular, okay, which is going to run right next to the lateral strut of your lower leg, the fibula. And then down here you've got the dorsalis pedis, which is a major landmark. You can take a pulse here. This is also a way to check for adequate blood flow to the lower limbs. Okay, so it's a nice diagnostic vessel. The plantar arteries run on the sole of the foot. The dorsalis runs on the top of the foot. Okay, these are showing you the principal veins, and long story short, essentially the venous route is very, very similar to the arterial route. It's just that the direction is different. There's a few exceptions, okay, and we'll talk about a few of them. Let's look at the vessels that drain the neck and the head, okay? So you have to think in terms of direction here. Essentially, blood coming from the brain enters the venous sinuses, okay, and drains into the vertebral, okay, there's the vertebral vein, and also into the jugular vein, okay, and the jugular vein has both an internal and an external branch, okay, this is the internal jugular shown here, and there is the external jugular shown here, all right. These jack into the subclavian, all right, and this jacks into the brachiocephalic, and which dumps directly now into the superior vena cava, which leads to the right atrium of the heart. The axillary also drains into this area. If we look at the upper limb, what we see here is these are the superficial veins, these are the deep veins, but essentially what are you picking up here? Well, the names are a little bit different in the forearm, okay? The superficial veins, these are the pulmar venous plexus. This drains blood from the palm of the hand. There is the median antebrachial vein, and this is the cephalic vein. Over here, the basilic, the median cubital. Okay, a lot of blood draws are taken here. The median cubital. Here is the right cephalic, the right basilic, and there is the axillary again, okay, under the arm, the brachiocephalic up here, the subclavian, and the right external jugular. And if we look at the deep veins, what we see are the radial and ulnar veins, the brachial veins, okay, and then we jack into the axillary, the subclavian, okay, and then we eventually join up with the jugulars, and then we jack into the superior vena cava and head back to the heart. These are the veins that feed the pelvis and the lower limbs. Okay. Again, this is anterior view. This is posterior view. So again, you have to think in terms of what's draining what. Okay. So here is the anterior tibial. Here is the saphenous. Okay, saphenous runs across the knee after it leaves the lower limb. These are the posterior tibials, run behind the tibia, and this is the greater saphenous up here. Okay, heading up, you've got the great saphenous, and eventually that is going to join up with the femoral. Okay, they will become now the right external iliac. It will join the internal iliac, and then it will become the common iliac, and then feed into the inferior vena cava. Posterior view, you get a better look at the fibular and the small saphenous, but essentially the root is the same. Okay, so that's just a few of the major vessels. Okay, let's talk about the pulmonary circulation. This is basically the route that the blood takes from the heart to the lungs and then from the lungs back to the heart. So it carries blood from the right side of the heart to the lungs to get oxygen and eliminate carbon dioxide. The route is from the right to the left. So we go into the right ventricle, to the pulmonary trunk, to the right and left pulmonary arteries, then to both lungs. From there, we carry deoxygenated or blue blood, which is low in oxygen and high in carbon dioxide, into the lungs. In the pulmonary capillaries, we have gas exchange, so we swap carbon dioxide for oxygen. What governs this primarily is the difference in concentration gradients of these two gases. Essentially, again, in the air that you breathe, oxygen concentration is high compared to the blood that's perfusing the lungs. So oxygen flows into the blood there. And carbon dioxide is high in the blood perfusing the lungs compared to the air that you're breathing. So carbon dioxide flows out of the blood perfusing the lungs and into the exhaled air. The right and left pulmonary veins then feed into the left atrium. This carries red blood, which is high in oxygen and low in carbon dioxide, back to the heart, and then we enter now the systemic circulation. There's a couple of special circulatory routes that we should address. These are called portal routes. The hepatic portal circulation is found in the liver. Okay, this transports blood from one organ's capillary bed to another. And this is what we mean when we say a portal circulatory route. In the GI organs, what happens is that you have splenic and superior mesenteric veins that eventually become now the hepatic portal vein. So there's deoxygenated blood in this vessel. What happens is that this joins the hepatic artery, and this feeds into sinusoids, which are large open spaces inside the liver. And in these sinusoids, what happens is that the blood has its chemistry adjusted. Essentially, there are detoxification processes that go on in the liver. The liver also adds serum albumin to the blood. It's also a major site of bile synthesis. Bile is secreted in the liver and does not enter the bloodstream, but it enters instead special bile ducts that eventually feed into the gallbladder, which, remember, is an accessory organ of digestion. The bile helps us to emulsify the fat that we eat so that we can more easily chemically break it down into its components, glycerol and fatty acids, which we then assemble into structures called chylomicrons, which enter lacteals, which are embedded in the walls of the GI. And once we're in the lacteals, we eventually rejoin the circulation at the subclavian veins. In these sinusoids, oxygenated and deoxygenated blood mix. The hepatic vein then drains the liver into the inferior vena cava. At this point now, the blood has had its nutrient content adjusted. That's another major activity that goes on in the liver. The liver basically has as one of its roles the body's kitchen. It decides what sort of fuel we want to prepare and put into the blood for delivery to other tissues. And so we adjust it on that basis. And then from there, we head out to the vena cava and eventually to the heart and to the rest of the body. And so this is just showing you the setup with the GI organs draining into the liver through the hepatic portal vein, okay, shown right there, the gateway to the liver. And then in the liver, all this cleaning and nutrient content adjustment goes on. And then we leave via the hepatic vein into the inferior vena cava. Okay, so this is a very important organ, the liver. Without it, we wouldn't be able to survive. This is just a diagram showing you the basic route to and from the liver. So if we start with the heart, the aorta delivers oxygenated blood, eventually making it to the abdominal aorta, which runs to the hepatic artery, which fuses with the hepatic portal vein, and now we enter the liver as a mixture of oxygenated blood and fuel-rich blood. Okay, uh, the hepatic portal vein collects blood from the stomach, the pancreas, the large intestine, as well as portions of the small intestine and the pancreas and the stomach. These drain into either the splenic or mesenteric vein, which empty into the hepatic portal vein. And so this blood is very rich in nutrients but very low in oxygen. It merges with this oxygenated blood, enters the liver. The liver then performs all the chemical processes necessary to adjust the fuel content and to detoxify whatever's in there. And then we send the blood out through the hepatic veins, which lead to the inferior vena cava, which make it back to the heart. Now, another special circulatory route is what takes place in the fetal circulation. And essentially, what happens in fetal circulation is that we don't have the benefit of breathing air. Okay? There's no air around. And so where do we get oxygen from? And the answer is we get it from mom. And the way we get it from mom is that we steal it from her blood through the placenta. Remember that the fetal blood and the maternal blood do not mix during pregnancy, but they do exchange wastes, nutrients, and gases. So what happens there is that deoxygenated blood essentially leaves the fetus, enters the umbilical cord through the umbilical artery, which carries blood away from the fetal heart, and then that deoxygenated blood in the placenta exchanges carbon dioxide and oxygen. It picks up oxygen, it dumps off carbon dioxide, it dumps off waste products and picks up nutrients, and then that blood enters now the umbilical vein, so it is now oxygen rich and nutrient rich, and then it returns back to the fetus. So this is a specialized system for the exchange of materials with the maternal blood bypassing the lungs. The exchange happens in the placenta via the umbilical vein. On the way back, okay, what happens here is that this oxygenated, nutrient-rich blood enters a special vessel called the ductus venosus, which runs on the underside of the liver and jacks directly into the inferior vena cava. This eventually brings this blood back to the right atrium, which results in a mixture of deoxygenated and oxygenated blood in the inferior vena cava of the fetus. Okay, the deoxygenated blood, remember, is making the return trip from the body, so it's already dumped off a lot of its oxygen and nutrient content. But when it meets the blood that's coming from the ductus venosus now, it gains oxygen and it gains nutrients. And then it enters now the right atrium. And in the right atrium, what we have is a very special structure called the foraminal valley, which is a bypass that allows blood to shunt from the right atrium to the left atrium. As a result now, when the left atrium squeezes, it can send blood into the left ventricle and then into the aorta, and that blood's going to be oxygenated. But there's another bypass, and it's called the ductus arteriosus, and it exists between the pulmonary trunk and the aorta. Okay, so when we squeeze blood out of the left ventricle in the fetus, and it enters the pulmonary trunk, it can also directly enter the aorta. I'm sorry, blood that enters the aorta in the fetus can directly enter the pulmonary trunk, and as a result, we can perfuse the lungs, even though they're not breathing air, with oxygenated blood. That allows them to develop. In addition to this, the right ventricle can pump blood to the, the right blood can pump blood to the ductus to the pulmonary trunk, then to the ductus arteriosus, into the aorta, into the internal iliac arteries, into the umbilical arteries, and then we head back to the placenta. And this is where we pick up now oxygen from mom and nutrients, and we dump off carbon dioxide and and waste products. Okay, so if we look at these different structures, you can see them here. Okay, this is the fetal circulation. This is the adult circulation. And what you want to notice is the ductus arteriosus. Okay, this little bypass right here that hooks the pulmonary trunk to the aorta. All right. The foramen ovale, which is between the right and the left atrium. The ductus venosus, which runs on the underside of the liver, okay, and brings blood back to the inferior vena cava. And down here, the umbilical vein, okay, which is carrying oxygenated blood back from the placenta into, again, the ductus venosus. And then down here, you've got the umbilical arteries. And what do the umbilical arteries do? They carry deoxygenated blood away from the fetus, and they send it directly into the um, umbilical artery that goes to the placenta. Okay. Now at birth what happens is that we breathe for the first time. And when we breathe for the first time, what happens is that all of these vessels, the ductus arteriosus, venosus, and the umbilical vein, they all collapse and become ligaments. So they no longer conduct blood. And the foramen ovale will close and become the fossa ovalis, which will be a small indentation in the inner atrial septum. This affects now a separation of the pulmonary and the systemic circulation. Now that the lungs are capable of taking in air, we don't have to rely on these bypasses to get oxygen into the fetal systemic circulation. And as a result now, we close them all off. Occasionally what can happen is that some of these structures can remain open at birth, and these are called patent fetal structures. And if, for instance, we leave the ductus arteriosus or the foramen ovale open at birth, what this will result in is a mixing of oxygenated and deoxygenated blood in the adult. And what this does is this decreases the efficiency with which we can deliver oxygen to metabolically active tissue. So this cuts down on your energy levels and on your activity levels. This can be surgically corrected if it's deemed necessary. So the changes that occur at birth are that the umbilical arteries become umbilical ligaments. The umbilical vein becomes the ligamentum teres. The ductus venosus becomes the ligamentum venosum. The placenta is expelled after birth. The foramen ovalis closes and becomes the fossa ovale. The ductus arteriosus closes and becomes the ligamentum arteriosum. And now the adult circulatory routes have been established. As we age, some of the effects on the circulatory system that decrease its efficiency include stiffening of the aorta, the loss of the strength of the cardiac muscle, which reduces cardiac output and systolic pressure, and the increased risk for coronary artery disease, congestive heart failure, and arteriosclerosis. And this comes primarily just from a lifetime of being exposed to your environment and also of using the heart, right? As you use the heart, what happens eventually is that it wears out because all tissue has a finite lifetime. And as you lose strength in the heart muscle, you lose cardiac output because the heart's weaker, it doesn't pump as much blood with every beat. And the result then is reduced cardiac output, reduced systolic pressure, and as a result now tissues aren't getting the oxygen, the nutrients they need in a timely fashion. The increased risk of coronary artery disease comes primarily from a slowed blood flow and also from the effects of arteriosclerosis. Essentially, what can happen is that we can narrow the lumen of these vessels and as a result increase their resistance. And this increases our blood pressure. And the negative effect that this can have is that it can cause capillary beds to rupture, and this can result in a stroke if it occurs in the brain, or a heart attack if it occurs in the heart muscle. Okay, so you might ask, well, where does this buildup inside the walls of these vessels come from? It comes from your diet. Okay, basically, plaques can accumulate on the walls of these arteries over time, especially if you have a diet that's high in saturated fat, cholesterol, and this is what contributes now to what we think of as coronary artery disease. And also, the arteries themselves can just inflame as a result of these deposits, and that further decreases their diameter. Congestive heart failure is basically uh, the heart um, stops beating or slows down to the point where it's not effective, and as a result, we do not affect circulation. Arteriosclerosis is one of the factors that can lead to coronary artery disease and also can lead to congestive heart failure because it increases peripheral resistance, increases the strain on the heart. It can also result in a condition known as cardiomyopathy, where the heart muscle can enlarge because it's having to do so much extra work. Okay, so all of these are negative effects resulting from aging on the heart and the cardiovascular system. Okay, that brings us to the end of this chapter. Uh, be sure, remember, and listen to the video podcast for chapter 22 and also chapter 24, which I'm going to post momentarily. Thanks.